Hey, it's Professor Gould, and we're finally ready to get to mitosis. I know, we got through G1, we got through S phase, we got through G2. We're going to pass the G2 checkpoint, and we're going to be able to start mitosis. Now, mitosis itself is a multi-step process. Remember that we're going to end up with two cells that have everything the first cell had. So G2 checkpoint is going to make sure we have duplicates of everything. And then mitosis is going to organize all of the cytoplasm, the organelles, and the chromosomes in particular, separate the sister chromatids, and separate the cytoplasm, and then make two cells. All right, so how do we do that? There were five phases of mitosis. The old prophase, metaphase, anaphase, telophase, cytokinesis, which I remembered by the acronym PMATSI, or Priscilla murdered another terrible clown. Why is she murdering clowns? I don't know. If that's too violent for you, maybe she married another terrible clown. Okay, but the problem is we've learned some more about this, and the people who name these things have decided that what we used to call late prophase should actually be considered a phase in and of itself, which we call prometaphase. So the new phases are prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. Sometimes considered like a whole separate thing, sometimes just an extension of telophase, whatever. Either way, interphase, totally separate, right? Not part of mitosis. Once we get past that G2 checkpoint, we're out of interphase and in the M phase of mitosis, okay? So this one is not part of it. All right. So we used to have, to remember these PMATSI, now we have PMAT, maybe a C? I don't know. It's not as great. Anyway, prophase, prometa, meta, anatelo. All right. During interphase, we can't see the individual chromosomes. Remember, they're in that heterochromatin structure. They're not condensed. What we do see in plant cells, we see these little dark spots. Those are nucleoli. Yes, plants have multiple nucleoli. Animal cells usually only have one. Why? You'd have to ask. It's beyond my knowledge of these things. How do we know this is a plant cell? It's got nice straight walls. The cell is kind of rectangular. Those are those cell walls made of cellulose. All right, in animal cells, which tend to be, they're usually kind of oval, although not always. We have a nice distinct nucleus, but we just have dark stuff in it. Like it's not really distinct. Once prophase starts, that's when the chromosomes condense, and now we can see them. So we can start to see these individual structures in the animal cells and individual chromosomes in the plant cell. The nuclear envelope is going to break down, and that's a really important part because remember, mitosis is going to separate everything in the cell, and we got to separate the sister chromatids and then put them in separate nuclei. They got to be in their own nucleus eventually. So we're going to break down the nuclear envelope so that we can organize everything. The cytoskeleton actually starts to break down, and structures called centrosomes, remember those centrosomes out in the cytoplasm, they're going to use those proteins from the cytoskeleton to make different microtubules called the spindle fibers. The centrosomes during prophase are going to start to move to opposite sides of the cell. We call those the poles. Okay, just like Earth has a north and south pole, a cell also has two poles at either side. So here's one chromosome telling another that hasn't condensed yet. Dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. Because you got to get condensed so that we can do prometaphase. All right. At prometaphase, the chromosomes continue to condense, and now they're really, really visible, really, really distinct. Okay, especially in the animal cells, but also in the plant cells, we can see very, very distinct individual chromosomes. The centrosomes move all the way to the end of the cells, and they keep making these spindle fibers. These spindle fibers are going to go all the way to the chromosomes, and then some of them are going to just overlap with others that are not connected to chromosomes. Each sister chromatid in this centromeric region develops what we call a kinetochore. This kinetochore is a set of proteins that are going to bind to these spindle fibers. Okay? So by the time we get to the end of prometaphase, the spindle fibers are made, the centrosomes are at the ends of the cell, and the spindle fibers are going to bind to the fibers of the kinetochore. All right, metaphase. Meta means next to. So metaphase is when all of the chromosomes are lined up next to each other, right? Now, we usually show them in a diagram lined up like this, but a lot of times in an actual stained cell, we see that the centers are lined up, but then like the legs of the chromosomes are kind of sticking out, okay? So the blue here are the chromosomes and the red are the spindle fibers. So to figure out what metaphase looks like in a cell, look for a line of chromosomes. And then sometimes we get lucky and we can actually see all of those spindle fibers, that like fan of spindle fibers. There's a centrosome here, and then you can see there's like more fibers going this way because there's a bunch of anchors and stuff to keep everything organized. Okay, so the spindle fibers are attached to the sister chromatids at those kinetochores. And then the other microtubules that aren't attached to the chromosomes, they're going to slightly overlap each other along this imaginary line called the metaphase plate, which is just an imaginary line that goes through the center of the cell between the two poles. These are called polar microtubules because they just extend from the pole to the metaphase plate or the equator. All right, so that's metaphase. So we did prophase, where the chromosomes started to condense and the nuclear envelope started to break down. We did prometaphase, where the spindle fibers got all made, the centrosomes moved to the end of the cells, and the nuclear envelope finally breaks down completely. Metaphase, we got everybody lined up, and now we're ready to separate the sister chromatids. Ooh, you know what? I forgot one thing I want to talk about back here. Centrosome is the organelle. Centromere is this area in the chromosome. And the only way I can remember which of these is which is that I remember that the centromere is merely a handle. Okay, it's just a handle to hold the two sister chromatids together and then to attach the chromatid to the spindle fiber. So the centrosome is a body out in the cytoplasm. Everything's cytoplasm at this point. Centromere is merely a handle on the chromosome. Okay, all right, moving on. We got metaphase. Next up, here's a close-up of what metaphase looks like. So you can see all the spindle fibers organized, attached to the sister chromatids that are lined up along this metaphase plate. Okay, checkpoint. Got to check yourself before you wreck yourself, because if you don't have metaphase organized correctly, if these spindle fibers aren't attached to these kinetochores on the sister chromatids, then separating them is not going to work. So there is a checkpoint here to make sure that the sister chromatids are correctly connected to those spindle fibers, which are microtubules. For those of you who are like, but which regulatory proteins are happening here? I don't know nor care. That's something for cell biology. Okay, so you will learn about that in cell biology, which will not be taught by me. Anaphase. The centrosomes are going to start shortening the spindle fibers. I know they just made them. Yeah, they make them, send them out, then pull them back in. As those spindle fibers shorten, the centrosome is going to break down, and the sister chromatids are going to be able to separate. And each sister chromatid gets pulled to a pole, and two sister chromatids get pulled to opposite poles. So we're going to end up with one full set of chromatids on one end of the cell, or chromosomes, and another set of chromosomes on the other end of the cell. Honestly, this is super elegant, and I love it. Again, this is really easy to identify in a real cell. So we can actually see the chromosomes and the little spindle fibers between them. Okay. The cell also begins to elongate, which is kind of hard to see a lot. It's not really distinctive until late anaphase, which looks a lot like telophase. Hello, this is telophase. Let me just go ahead and fix that right now. Like, honestly, I just can't with myself right now. This is telophase. Now it's fixed. Okay. Telophase. everything was happening exactly the same, whether we were in a plant or an animal cell, but right now we're going to have to do something different. Animal cells are going to do this slightly different than plant cells, and that's because animal cells are going to actually fully separate from each other, whereas the plant cells are going to be stuck together with a cell wall between them. All right, so during telophase, the spindle fibers get broken down, cytoskeleton gets remade, and a special part of cytoskeleton gets made around this metaphase plate. Okay, so we're going to see like a new line appearing, and in animal cells, we're going to see a cleavage furrow. Stop staring at my cleavage. Okay, so this is what it looks like in an animal cell. We have a set of microtubules that get created here at the metaphase plate, and they actually start to pinch the plasma membrane, and they create this cleavage furrow. A cleavage is just a separation between two things. There you go. That ring keeps contracting until it completely pinches the plasma membrane and creates two new cells. These are called daughter cells. So the daughter cells, at that point that the daughter cells have been created, cytokinesis has happened. So if you're wondering, like, where is the dividing line between telophase and cytokinesis, the answer is it's not like super, I mean, it's kind of when the new cells are created, but some people call this whole pinching thing cytokinesis, so it's not super clear. Cyto means cell. There's that kine again, which means movement, so literally cells moving apart from each other. Now, some people say, oh, yeah, once this has happened, the cells are separate, and we are never, ever, ever getting back together, because they're totally separate cells now. Apologies to Taylor. Okay, now, in plant cells, they don't move apart from each other. They do get separated by a cell wall, but they don't, like, move away from each other. So some people say plants don't do cytokinesis, but like we don't have another great term for it, so I just call it cytokinesis. A lot of people call it cytokinesis, so we're just going to call it cytokinesis. All right. So we do make up a set of microtubules at the metaphase plate, just like in animal cells. We also have a set of vesicles from the Golgi apparatus that arrive at this place, and those create, the microtubules and the vesicles create this thing called a phragmoplast, which is just an awesome name, phragmoplast. Those all then, the vesicles all fuse together to create a cell plate. And the cell plate is just, it'll grow up to be a cell wall, but it hasn't reached the plasma membrane yet. Once it reaches and fuses with the plasma membrane, then it grows up into be a cell wall. Okay, so we have phragmoplast, which is microtubules. They attract the vesicles from the Golgi. They all fuse together into a cell plate, which then keeps growing into a cell wall. And now we have two distinct plant cells. And we are done with mitosis in both the animal and plant cells. And honestly, we have to stop talking about mitosis. It's such a divisive issue. All right, I'm going to have one more little section for you about how prokaryotic cells do this process, because they can't do mitosis. They only have a single round chromosome, so they don't have to do quite all of this. And also they don't have a nuclear envelope, so they're not going to break down nuclear envelope, are they? Ooh, I should mention that. The other thing that happens during cytokinesis is that the nuclear envelope reforms during telophase. Not during cytokinesis, during telophase, the nuclear envelope reforms. So we have two cells with a full complement of chromosomes in their nucleus. And that, my friends, is mitosis.